Now we have chapter 15, class 11, NCERT Body Fluid and Circulation. You have learned that all living cells have to be provided with nutrients, oxygen and other essential substances. Also the waste or harmful substances produced have to be removed continuously for healthy functioning of tissue. It is therefore essential to have efficient mechanism for the movement of these substances to the cells and from the cells. Different groups of animals have evolved different methods for the transport. Simple organisms like sponges and coelacanths circulate water from their surroundings through their body cavities to facilitate the cells to exchange these substances. More complex organisms use special fluids within their bodies to transport such materials. Blood is the most commonly used body fluids by most of the higher organisms, including humans, for this purpose. Another body fluid, lymph, also helps in transport of certain substances. In this chapter, you will learn about the composition and properties of blood and lymph tissue fluid, and mechanism of circulation of blood is also explained herein. Now, blood is a special connective tissue consisting of a fluid matrix, plasma and formed elements. Plasma is a straw colored viscous fluid constituting nearly 55% of the blood. 90 to 92% of the plasma is water and protein contribute 6 to 8% of it. Fibrinogen, globulin, albumins are the major protein. Fibrinogens are needed for clotting or coagulation of blood. Globulins primarily are involved in defense mechanism of the body, and albumins helps in osmotic balance. Plasma also contains small amounts of minerals like sodium, calcium, magnesium, HCO3 and chlorine, etc. Glucose, amino acid, lipids, etc. are also present in plasma as they are always in transit in the body. Factors for coagulation or blood clotting are also present in plasma in an inactive form. Plasma without the clotting factor called serum. Formed element. Erythrocytes, leukocytes and platelets are collectively called as formed elements, and they constitute nearly 45% of the blood. Erythrocytes or red blood cell are the most abundant of all the cells in the blood. A healthy adult man has on an average 5 million to 5.5 million of RBC per mm³ of blood. RBC are formed in the red bone marrow. In the adults, RBC are devoid of nucleus in most of the mammals and are biconcave in shape. They have a red colored iron containing complex protein called hemoglobin, hence the color and the name of these cells. A healthy individual has 12 to 16 gram of hemoglobin in every 100 ml of blood. These molecules play a significant role in transport of respiratory gases. RBCs have an average lifespan of 120 days, after which they are destroyed in the spleen, graveyard of RBC. Leukocytes are also known as white blood cells. As they are colorless due to lack of hemoglobin, they are nucleated and relatively lesser in number, which averages 6,000 to 8,000 mm³ of a blood. Leukocytes are generally short-lived. We have two main categories of WBC, granulocytes and agranulocytes. Neutrophils, eosinophils and basophils are different types of granulocytes, while the lymphocytes and monocytes are the agranulocytes. Neutrophils are the most abundant cells, 60 to 65% of the total WBC, and basophils are the least, 0.5 to 1% among them. Neutrophils and monocytes, 6 to 8%, are phagocytic cells which destroy foreign substance entering the body. Basophils secrete histamine, serotonin, heparin, etc. and are involved in inflammatory reaction. Eosinophils 2 to 3% and resist infection and are also associated with allergic reaction. Lymphocytes 20 to 25% are of two major types, B and T forms. Both B and T lymphocytes are responsible for immune response of the body. Platelets, also called thrombocytes, are cell fragments produced from megakaryocytes, special cells in the bone marrow. Blood normally contains 1.5 lakh to 3,50,000 platelets per mm³. Platelets can release a variety of substances, most of which are involved in the coagulation of clotting of the blood. A reduction in their number can lead to clotting disorder, which will lead to excessive loss of blood from the body. Blood group. As you know, blood of human differs in certain aspect, though it appears to be similar. Various type of grouping of blood has been done. Two such grouping, the ABO and RH negative, are widely used all over the world. ABO grouping. ABO grouping is based on the presence of or absence of two surface antigen chemicals that can induce immune response on RBC, namely A and B. Similarly, the plasma of different individuals contain two natural antibodies, protein produced in the response to antigen. The distribution of antigen and antibodies in the four groups of blood A, B, AB, and O are given in table. You probably know that during blood transfusion, any blood cannot be used. The blood of a donor has to be carefully matched with the blood of the recipient before any blood transfusion to avoid severe problems of clumping, destruction of RBC. The donor's compatibility is also shown in Figure 1 table. Here is the table. From the above mentioned table, it is evident that group O blood can be donated to person with any other blood group, and hence O individual are called universal donor. Person with AB group can accept blood from person with AB as well as other groups of blood. Therefore, such persons are called universal recipients. RH grouping. And another antigen, the antigen similar to one person is the rhesus monkey, hence RH, is also observed on the surface of RBC of a majority, nearly 80% of humans. Such individuals are called RH positive, and those whom this antigen is absent are RH negative. An Rh negative person, if exposed to Rh positive blood, will form specific antibodies against Rh antigen. Therefore, Rh group should also be matched before transfusion. A special case of Rh incompatibility has been observed between the Rh negative blood of a pregnant mother with the Rh positive of the fetus. Rh antigen of the fetus do not get exposed to the Rh negative blood of the mother in the first pregnancy, as the two bloods get separated by placenta. However, during the delivery of the first child, there is a possibility of exposure of maternal blood to small amounts of Rh positive blood from the fetus. In such case, the mother starts preparing antibodies against Rh antigen in her blood. In case of her subsequent pregnancy, the Rh antibodies from the mother can leak into the blood of the fetus Rh positive and destroy the fetal RBC. This could be fatal to the fetus or could cause severe anemia and jaundice to the baby. This condition is called erythroblastosis fetalis. This can be avoided by administering anti-Rh antibodies to the mother immediately after the delivery of the first child. Now, coagulation of blood. You know that when you cut your finger or hurt yourself, your wound does not continue to bleed for a long time. Usually the blood stops flowing after some time. Do you know why? Blood exhibits coagulation or clotting in response to an injury or trauma. This is a mechanism to prevent excessive loss of blood from the body. You would have observed a dark reddish brown scum form at the site of a cut or injury over a period of time. It is a clot or coagulum formed mainly of a network of threads called fibrins, in which the dead and damaged foam elements of the bloods are trapped. Fibrins are formed by the conversion of inactive fibrinogen in the plasma by enzyme thrombin. Thrombins in turn are formed from the another inactive substance present in the plasma called prothrombin. An enzyme complex, thrombokinase, is required for the above reaction. This complex is formed by a series of linked enzymatic reaction, cascade process, involving a number of factors present in the plasma in an inactive state. An injury or a trauma stimulates the platelets in the blood to release certain factors which activates the mechanism of coagulation. Certain factors released by the tissue at the site of injury also can initiate coagulation. Calcium ions play a very important role in clotting. Lymph, the tissue fluid. As the blood passes through the capillaries in tissue, some water along with the many small water-soluble substances move out the space between the cells of the tissue, leaving the larger proteins and most of the foam elements in the blood vessels. This fluid released out is called the interstitial fluid or tissue fluid. It has the same mineral distribution as that in plasma. Exchange of nutrients, gases, etc. between the blood and the cells always occur through this fluid. An elaborate network of vessels called the lymphatic system collects this fluid and drains it back to the major veins. The fluid present in the lymphatic system is called the lymph. Lymph is a colorless fluid containing specialized lymphocytes which are responsible for the immune responses of the body. Lymph is also an important carrier for nutrients, hormones, etc. Fats are absorbed through the lymph in the lacteals present in the intestinal villi. Circulatory pathway. The circulatory pattern of the two types, open or closed. Open circulatory system is present in arthropods and mollusks, in which blood pumped by the heart passes through the large vessels into open space or body cavities called sinuses. Annelids and chordates have a closed circulatory system in which the blood pumped by the heart is always circulated through a closed network of the blood vessels. This pattern is considered to be more advantageous as the flow of blood can be more precisely regulated. All vertebrates possess a muscular chamber heart. Fishes have a two-chambered heart with atrium and ventricular. Amphibians and the reptiles except crocodile have a three-chambered heart with two atria and a single ventricular, whereas crocodiles, birds, mammals possess a four-chambered heart with two atria and two ventricles. In fishes, the heart pumps out deoxygenated blood, which is oxygenated by the gills and supplied to the body parts, from where the deoxygenated blood is returned to the heart. In amphibians, the reptiles, the left atrium receives oxygenated blood from the gills, lungs, skins, and the right atrium gets deoxygenated from other body parts. However, they get mixed up in the single ventricle, which pumps out mixed incomplete blood, double circulation. In birds, mammals, oxygenated and deoxygenated bloods received by the left and right atria respectively passes onto the ventricle of the same side. The ventricles pump it out without any mixing up, that is, two separate circulatory pathway present in these organisms. Hence, these animals have double circulation. Let us study the human circulatory system. Human circulatory system, also called the blood vascular system, consists of muscular chamber heart, a network of closed branching blood and blood vessels and blood, the fluid which is circulated. Heart, the mesodermally derived organs, is situated in the thoracic cavity in between the two lungs, slightly tilted to the left. It has the size of a clenched fist. It is protected by a double-walled membranous bag, pericardium, enclosing the pericardial fluid. Our heart has four chambers, two relatively small upper chambers called atria and two large lower chambers called ventricles. A thin muscular wall called the interatrial septum separates the right and the left atria, whereas thick-walled the interventricular septum separates the left and right ventricular. The atrium and the ventricle of the same side are also separated by a thick fibrous tissue called atrioventricular septum. However, these septa are provided with an opening through which the two chambers of the same side are connected. The opening between the right atrium and the right ventricle is guarded by a valve form of the three muscular flaps or cusp. The tricuspid valve, where a bicuspid or mitral valve guards the opening between the atrium and the left ventricles. The opening of the right and the left ventricles into the pulmonary artery and the aorta respectively are provided with the semilunar valves, and the valves in the heart allows the flow of the blood only in one direction, that is from the atria to the ventricles, and from the ventricles to the pulmonary artery or aorta. These valves prevent any backward flow. The entire heart is made up of cardiac muscles. The wall of the ventricles and are much thicker than of atria. A specialized cardiac musculature called the nodal tissue is also distributed in the heart. A patch of this tissue is present in the right upper corner of the atrium called sinoatrial node, and another mass of the tissue is seen in the lower left corner of the right atrium close the atrioventricular septum called the atrioventricular node. A bundle of nodal fiber, atrioventricular bundle, continues from the AVN, which passes through the atrioventricular septa to emerge on the top of the interventricular septum and immediately divides into the right and the left bundle. These branches give rise to the minute fibers throughout the ventricular musculature of the respective side of are called Purkinje fiber. The nodal musculature has the ability to generate action potential without any external stimuli, that is, it is auto-excitable. However, the number of action potential that could be generated in a minute vary at different parts of the nodal system. The SAN can generate the maximum number of action potential, that is 70 to 75 per minute, and is responsible for initiating and maintaining the redeeming contractile activity of the heart. Therefore, it is called the pacemaker. Our heart normally beats 70 to 75 times in a minute, average 72 beats. Cardiac cycle. How does the heart function? Let us take a look. To begin with, all the four chambers of the heart in a relaxed state, that is, they are in joint diastole, as the tricuspid or bicuspid valve are open from the pulmonary veins and vena cava flows into the left and right ventricle respectively through the left and right atria. The semilunar valves are closed at this stage. The SAN now generate an extend potential which stimulates both the atria to undergo a simultaneous contraction. The atrial systole, this increased the flow of the blood into the ventricles by about 30%. The action potential is conducted to the ventricular side by AVN and AV bundle, from which the bundle of is transmit it through the entire ventricular musculature. This causes the ventricular muscles to contract. The atria undergo relaxation diastole coinciding with the ventricular systole. Ventricular systole increases the ventricular pressure, causing the closure of tricuspid and bicuspid valves due to attempt backflow of the blood into the atria. As the ventricular pressure increases further, the semilunar valves guarded the pulmonary artery and the aorta are forced open, allowing the blood in the ventricles to the flow through these vessels into the circulatory pathway. The ventricles now relax and ventricular pressure falls, causing the closure of semilunar valves, which prevents the backflow of blood into the ventricles. As the ventricular pressure declines further, the tricuspid and bicuspid valves are pushed open by the pressure in atria exerted by the blood, which was being emptied into them by the veins. The blood now once again moves freely to the ventricles. The ventricles and atria are now again in relaxed state as earlier. Soon the SAN generates a new action potential and the events described above the repeated in that sequence and process continues. This sequence events in the heart, which is cyclically repeated, is called the cardiac cycle, and it consists of systole and diastole of both the atria and ventricles. As mentioned earlier, the heart beats 72 times per minute, that is, many cardiac cycles are performed per minute. From this, it could be deduced that the duration of the cardiac cycle is 0.8 seconds. During a cardiac cycle, each ventricle pumps out approximately 70 ml of blood, which is called the stroke volume. The stroke volume multiplied by the heart rate number of beats per minute gives the cardiac output. Therefore, the cardiac output can be defined as the volume of the blood pumped out by each ventricle per minute and average 5000 ml or 5 liter in a healthy individual. The body has the ability to alter the stroke volume as well as the heart rate and thereby cardiac output. For example, the cardiac output of an athlete will be much higher than that of an ordinary man. During each cardiac cycle, two prominent sounds are produced which can be easily heard through a stethoscope. The first heart sound, love, is associated with the closure of tricuspid and bicuspid valves, whereas the second heart sound, dove, is associated with the closure of the semilunar valves. These sounds are clinical diagnostic significance. The ECG, electrocardiograph. You are probably familiar with this scene from a typical hospital television show. A patient is hooked up with a monitoring machine that shows the voltage trace on a screen and make the sounds beep, 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 beep as the patient goes into the cardiac arrest. This type of machines, electrocardiograph, is used to obtain an electrocardiogram. Electrocardiogram is a graphical representation of the electrical activity of the heart during cardiac cycle. To obtain the standard ECG, a patient is connected to the machine with the three electrical leads, wrist and ankle, and that continuously monitor the heart activity. For a detailed evaluation of the heart's function, multiple leads to the attached to the chest region. Here we will talk about only a standard ECG. Each peak in the ECG identified with the letter to form P to T corresponds to a specific electrical activity of the heart. The P wave represents the excitation or depolarization of the atria, which leads the contraction of both the atria, and the QRS complex represents the depolarization of the ventricles, which initiates the ventricular contraction. The contraction starts shortly after Q marks and beginning of systole. The T wave represents the return of the ventricles from excited to normal, obviously. By counting the number of QRS complexes that occur in a given time period, one can determine the heartbeat rate of individuals. Since the ECG obtained from the different individuals have roughly the same shape for the given lead configuration, any deviation from this shape indicates a possible abnormality or disease. Hence, it is of a great clinical significance. Double circulation. The blood flows restricted by fixed route through the blood vessels, the arteries and the veins. Basically, each artery and veins consisting of three layers: the inner layer lining of squamous epithelium, endothelium and tunica intima, and a middle layer of smooth muscles and elastic fiber, the tunica media, and an external layer of fibrous connective tissue with collagen fiber, and the tunica externa. The tunica media is compact comparatively thin in the veins. As mentioned earlier, the blood pumped by the right ventricle enters the pulmonary artery, whereas the left ventricle pumps blood into the aorta. The deoxygenated blood pumped into the pulmonary artery is passed on to the lungs, and whereas the deoxygenated blood is carried out by the pulmonary veins into the left atrium. This pathway constitutes the pulmonary circulation. The oxygenated blood entering the aorta is carried by a network of arteries, arterioles, and capillaries to the tissues, from where the deoxygenated blood is collected by a system of venules. Veins and vena cava are emptied into the right atrium. This is the systematic circulation. Systematic circulation provides nutrient, oxygen, and other essential substances to the tissue and makes CO2 and the other harmful substance away from the elimination. A unique vascular connection exists between the digestive tract and liver called hepatic portal system. The hepatic portal veins carries blood from intestine to the liver before it delivered to the systematic circulation. A special coronary system of the blood vessels is present in our body exclusively for the circulation of blood to and from the cardiac musculature. Regulation of cardiac activity. Normal activities of the heart are regulated intrinsically, or that is auto-regulated by specialized muscles, nodal tissues. Hence the heart is called myogenic. A special neural center in the medulla oblongata can moderate the cardiac function through autonomic nervous system. Neural signals through the sympathetic nerve parts of ENS can increase the rate of the heartbeat, of the strength of the particular contraction, and thereby the cardiac output. On the other hand, parasympathetic neural signals, another component of ANS, decrease the rate of the heartbeat, speed of conduction of action potential, and thereby the cardiac output. Adrenal medullary hormones can also increase the cardiac output. Disorder of the circulatory system. High blood pressure, hypertension, is the term for the blood pressure that higher than normal, 120 by 80. In this measurement, 120 mmHg is systolic or pumping pressure, and the 80 mmHg is diastolic or resting pressure. If repeated checks of the blood pressure of individuals 140 by 90 or higher, it shows hypertension. High blood pressure leads to the heart disease and also affect vital organs like brain and kidney. Coronary artery disease. Coronary artery disease, often referred to as the atherosclerosis, affect the vessels that supply blood to the heart muscles. It is caused by deposit of calcium, fats, cholesterol, and fibrous tissue, which makes them lumen of arteries narrower. Angina. It is called the angina pectoris. A symptom of acute chest pain appears when no enough oxygen is reaching the heart muscles. And angina can occur in men and women of any age, but it is more common among the middle aged and elderly. It occurs due to conditions that affect the blood flow. Heart failure. Heart failure means the state of heart when it is not pumping blood effectively enough to meet the needs of the body. It is sometimes called congestive heart failure because congestion of the heart lung is one of the main symptoms of this disease. Heart failure is not the same as the cardiac arrest when the heart stops beating, or heart attack when the heart muscles is suddenly damaged by the inadequate blood supply. So that's it. Thank you so much.